I'd like to welcome you all to the fifth webinar in our Let's Talk MPTs discussion series. For those who do not know me, I'm Bethany Young-Holt, and I direct the initiative for MPTs and CAMI Health, and we're housed at the Public Health Institute. So as people continue to join, I want to just go over a few housekeeping points. We want to note that everyone will be muted during the presentations, and then we will unmute everyone during the Q&A. If you have questions for our discussants during their presentations, please do use the chat. For technical issues, you can chat my colleague Susanna Moore, who is our tech host today. Also, the webinar recording and related resources will be available on our website at theiMPT.org soon after the webinar. So today's Let's Talk MPTs webinar discussion will provide an overview of opportunities and challenges for MPT and HIV prevention drug delivery approaches. I'm so excited to have presentations today by two product developers who are working on really innovative drug delivery technologies that are being explored for HIV PrEP and MPTs. Dr. Rahima Binhabor from University of North Carolina, Chapel Hill will walk through her work that looks at the next generation 3D printed intravaginal rings as multi-purpose prevention technologies for prevention of HIV and unplanned pregnancies. Next, Courtney Jahan from PATH will describe their work on dissolving microarray patches for long-acting HIV and pregnancy prevention MPTs. Their presentations will be followed by a facilitated discussion and Q&A led by the IMPT's longstanding scientific advisor, Dr. Joe Romano. So we encourage you again to submit your questions in the chat feature of Zoom during your presentations, and we'll monitor these so that they can be answered during the Q&A. And you can also ask questions directly during the Q&A. So over 24 MPTs are currently in development with active funding that aim to provide prevention from 2 or more risks: unintended pregnancy, HIV, and or other sexually transmitted infections. MPTs have the potential of revolutionizing health and well-being by incorporating prevention of HIV and other STIs into contraceptives, which are less stigmatized, and reduce the burden of multiple clinic visits, the importance of which has been all the more recognized amidst the global COVID pandemic. But there are many complexities in developing MPTs that will be safe, effective, affordable, and used by women. So key challenges for MPT development include navigating the regulatory, CMC, toxicity, and other issues involved when combining different active pharmaceutical ingredients, or APIs. Furthermore, understanding the desirability and acceptability of an MPT candidate during its developmental stages so that when it reaches the market, MPTs will be used by women is also highly critical. So importantly, this includes developing appropriate delivery forms for MPTs that meet the diverse needs of women throughout their lifetime and for women who live in different geographical regions. So to help unpack some of these challenges and opportunities, we launched the Let's Talk MPTs discussion series around 10 months ago to engage and connect experts from diverse disciplines around the globe to highlight key issues, recognizing that these 60 to 90-minute discussions are only really the tip of the iceberg for many of these topics. But through these discussions, we aim to connect multidisciplinary thought leaders and further stimulate innovation and collaboration. Past topics in this series include the role of cervical vaginal microbiota on contraception, HIV prevention and MPTs, the integration of social behavioral and market research on MPT R&D, toxicity study considerations for MPT development, and combining APIs for HIV prevention and MPTs. And today we are really excited for our discussion on diverse delivery types for MPTs. So all of these webinar recordings are available on our website if you go to the Learning Events Hub at theimpt.org. So the MPT product pipeline is dynamic with candidates entering and leaving. Among the activities that the Initiative for MPTs does is to track the MPTs in a database. As of June, so just a couple weeks ago, there are 27 candidates in development with active funding representing an array of delivery types. The MPT delivery types technologies actually leverage the delivery technologies for other disease areas, including TB, influenza, rabies, contraception, and most recently advances in HIV prevention. Social behavioral research has demonstrated that women want choice to help make informed decisions about about their health. And this is especially critical for young women and adolescent girls who, due to multiple factors, including sexual violence and barriers to accessing contraception, are at high risk for unintended pregnancy. So it's really exciting to see this pipeline diversify. So today's discussion is going to focus on two innovative MPT delivery approaches in early stages of development: 3D printed intravaginal ring technologies and microarray patches, also known as microneedles. And I am excited to welcome our featured discussants, each of whom brings a wealth of expertise. So Rahima Binhabor is currently a tenure-track assistant professor at UNC in the Joint Department of Biomedical Engineering with a secondary appointment in the Esselman School of Pharmacy. She is the founder and director of Anelio Incorporated, a UNC spin-off company using the innovative 3D printed intravaginal ring platform technology for unmet women's health indications and as next generation MPTs. She received her BS in polymer engineering from the Algerian Petroleum Institute, a master's in medicinal organic chemistry at the University of Minnesota Duluth, and a PhD in organic polymer chemistry at McMaster University. She is also the inventor of in 5 patent applications and has earned multiple awards for her work. Courtney Jerian oversees a portfolio of projects at PATH, advancing vaccine and pharmaceutical packaging and delivery technologies, and is the co-leader of the Microarray Patch Center of Excellence at PATH. In collaboration with private and public sector partners, Courtney implements projects to develop, assess, and introduce combination products and medicinal devices with a focus on user-centered design, technical development, human factors evaluation, health economics, manufacturing, regulatory pathways, and commercialization. And a key area of focus is advancing the microarray path technology platform for delivery of essential medicines in low resource settings, including HIV PrEP and contraception. And finally, Joe Romano is president of the NWJ Group, a biotechnology and pharmaceutical consulting firm that focuses primarily on nonprofit and academic drug support for drug, device, and diagnostic development, and consults to multiple organizations involved in HIV prevention product development. Joe has also been a scientific advisor to the Initiative for MPTs since our inception around 12 years ago. So prior to his role at NWJ Group, Joe was the chief of product development at the International Partnership for Microbicides, has had a lead role at Biosyn, and spent 10 years at ABL Incorporated, where he was involved in research related to Kaposi sarcoma vaccine development. So Dr. Romano's training is primarily in molecular biology, immunology, and he received his PhD in microbiology from Rutgers. So with that, I am going to hand it over to Rahima. Thank you.
Thank you, Bethany, for that kind introduction. And hi, everyone. It's good to see so many wonderful people signed in. Let me just go ahead and share. Can you see the slides? Okay. Okay. So as Bethany said, today I'm going to focus on one of the couple technologies that we're developing for HIV prevention, but also as multi-purpose prevention technologies. And I'm going to be presenting on our 3D printed intravaginal ring technology that we're currently developing as an MPT. These are my disclosures. So I just wanted to start with just giving an overview of the current landscape of HIV prevention and contraception, just leading the way to why developing MPTs. So as many of you know, in terms of HIV prevention, currently the only preventative method is Truvada and Descovy as daily oral pills. Cabela is advancing into phase 3 clinical trials, and that's the long-acting injectable being developed by ViiV. For long-acting contraception, there are a couple long-acting contraceptives, such as Depo-Provera, which is an injectable. So as an injectable for long-acting, Cabanova has recently been approved as a treatment for HIV, which is a combination of cabotegravir and rilpivirine, which is very exciting. And in terms of long-acting implants, we've got the intravaginal rings. So for a contraception, this is a 1-year contraceptive, Annovera, which was recently approved. And Merck is developing their islatravir implant based on their Nexplanon technology for long-acting prevention of HIV. And so why is it so important to develop MPTs? And it is so exciting to see that there are so many different technologies that are currently in the pipeline. And that's because of these staggering statistics. I love this slide by IMPT, which I always kind of like steal from you guys, but it really sums it up all. With all these staggering statistics for both unplanned pregnancies, but also for HIV. And so, as you can see, 500,000 of young women are infected with HIV every year. So it is really important to look at various ways of developing a multi-purpose prevention technology that could be acceptable, but also accessible to women and to a larger population. So for today, as I said, I'm only going to focus on presenting on our 3D printed intravaginal ring technology that's being developed as an MPT with 2 different MPTs in development currently. And so just to introduce the technology that we're using, we are harnessing the high-speed, state-of-the-art 3D printing technology known as continuous liquid interface production. And in this technology, unlike the traditional layer-by-layer 3D printing technologies that have existed for a couple decades already, CLIP really is an ingenious technology because it's a continuous process that allows for fabrication of parts at very high speeds that can lead to the ability to commercialize and scale at the commercial scale for production, but also produce parts with very high resolution, which is very important when developing a product like a multi-purpose technology or an intravaginal ring that's an implantable device for women's health. And so the way this technology works is it's actually, as I said, it's a very neat technology because it takes a counterintuitive process in polymer chemistry by using oxygen to inhibit polymerization right above this oxygen permeable window to create what we call a dead zone. So within that dead zone, the resin material stays unpolymerized, so it remains liquid. And above the dead zone, where there is no oxygen, that's where the polymerization happens, and you get this continuous process that allows fabrication at high speed that can be scaled to commercial production. And so because of the nature of the technology, there are various knobs and components that we can change and optimize when 3D printing parts. These can include the speed by which the build platform is being pulled, or the material is being pulled upwards when in the fabrication process. We can actually optimize the resin properties, the dead zone thickness, the light intensity that's projected to fabricate, so on. And so this is just to give you kind of like a view of what a 3D printed interventional ring process looks like. So this is a sped up process for printing at a lab scale. This is printing 16 rings at once. As I said, this is a lab scale production. When we can print both horizontally and vertically, and when we print vertically, we can increase the throughput. The picture on the left is a 3D printing. The video on the right, excuse me, is the light projection that allows us to follow in real time the 3D printing process, and we can actually capture what's going on, but also get a lot of information in terms of print properties and the part properties while it's going in real time. And so, as I mentioned, this is the ability to use computer-aided design allows us not only print same design in a batch of several rings per batch, but also we can actually have different designs that can be printed all at once. It doesn't have to be the same design. And so initially, we were exploring printing, the differences between printing horizontally versus vertically. There's really no difference in terms of the part properties, except for we're increasing the throughput by significantly reducing the time per ring that we can print when we move to a vertical print process. We have also implemented IDs that we incorporate for every single ring. So these are imprinted that allows us to QC these rings and follow each ring with their ID in terms of characterization and performance. And so how does our technology work as a general overview for how we design these rings? We start with this torus, simple CAD design. We implement what we call the band. The torus cylindrical CAD design is where we embed our unit cells or the design. So we call these unit cells, and these can have any different shapes, sizes, and then the band that we can put around the ring is what allows for that smoothing of the ring and the ability to actually control the mechanical properties independently from controlling the release kinetics. So this was really critical for us because by doing so, we're able to precisely fine tune and control the release kinetics of drugs with the designs that we put in, the unit cell, and we're able to independently control the compression and the mechanical properties, which we know are very important from an acceptability standpoint using the band. And this was actually detailed in this publication on how we actually do that. So we've actually designed a library of different designs and tested these designs with different drugs with different properties to allow us to kind of like build a library, kind of like a combinatorial library of how the design aspects influence drug release kinetics and how we can actually start kind of understanding the different properties of a drug, the interaction between the correlation of properties of a drug, the resin material, the design of the ring, and how we get the release kinetics so that we can start building, reducing the time for R&D and increasing the throughput for precise drug delivery with different drugs and APIs. In terms of the surface area, as you can see here, this is the surface area of an S ring and a NUVA ring. These are the injection molded rings without any designs. So because we're implementing computer-aided designs and 3D printing, we have the ability to introduce these designs that cannot otherwise be introduced by injection molding. What that does is, as you can see here, it significantly enhances the surface area of these rings, which gives us that significant control and unique control over the drug release kinetics and duration for particular APIs. And so, as I mentioned, we wanted to control the mechanical properties of these rings in a way that would not interfere with the way we control the drug release kinetics. And what that does is gives us the ability to produce drug-loaded rings that for the same that can lead to the same release kinetics for a particular API with rings that can have different compressions. And that is allowed by the band properties, as I mentioned, going from a small band all the way to a ring that's completely covered. And that allows us to have mechanical properties that are cover a much wider area or range in compression compared to the S ring and NUVA ring shown here. So these, we always use the marketed products as our benchmarks, and you can see that we can hit compression forces that are either higher or lower than the marketed product just by changing the properties of these, of the band. So looking at how design properties changes or influences drug release kinetics, we took a model drug, but then we expanded it to multiple other drugs to develop this algorithm. So this is just data showing drug release kinetics that were obtained for beta-estradiol using different designs. And as you can see, just by changing the design, we're able to release the same drug anywhere from a few weeks to all the way to several months. One really important aspect about these 3D printed rings as well is the ability to achieve near complete release of the APIs that we're loaded in these rings, which is not actually achievable with injection molded rings. And based on the data that we get from the various designs, we were able actually to find a correlation between the specific surface area of these rings and the drug release kinetics and develop an algorithm that we could then use to go back and look at our library if we were to develop a ring for a particular drug that has similar properties as the number of drugs to which we use to develop this algorithm. We would know exactly which design to at least pick to start developing that particular ring for an API, given the product portfolio or the indication that we're trying to hit. We also developed a novel drug loading process that, given the importance of looking ahead in terms of regulatory approval, it was very important for us to completely disassociate the drug loading process from the 3D printing process, just to minimize the hurdles in terms of exposing an API to UV exposures and radical chemistry and thermal post-cure. And by doing so, and by dissociating the drug loading process from the fabrication process, this actually allows us, opens the door to the ability to formulate drugs that are otherwise sensitive to any heat or pressure that are inherent to the injection molding process, but also to UV exposure, which is inherent to our 3D process. So we can actually load biologics, proteins, antibodies or molecules that are sensitive to heat and pressure or UV exposure. So we, of course, tested these rings. We did a lot of in vitro and in vivo safety studies, both in mice and in the sheep, and in non-human primates, looking at different cell lines that are relevant for vaginal delivery. But also in collaboration with Rick Piles at UTMB, we looked at the safety of these rings in an ex vivo vaginal microbiome cell line that they developed. And also looked at histology for implantation sites in mice and in the sheep. In the sake of time, I'm not going to show the sheep data, I'm going to show a little bit of the development of the two MPTs that are currently in development. One to deliver an anti-HIV, dapivirine, an anti-HSV, pritelivir, and a contraceptive, levonorgestrel. So this is the first triple drug MPT that we're developing. This was a work funded by NIH and IAID. So this is just a picture showing the design that we locked in for this particular MPT. And this is a human size ring compared to the non-human primate ring, compared to the tiny little mouse ring, which we can actually, so the metrics for the human ring to the macaque ring were actually kept constant. So we made sure that the specific surface area and all the diffusion distances were kept constant, going from the human size to the macaque size, so we can do a comparison between the sheep PK and the non-human primate PK. So as I mentioned, this was funded by an R61 grant from NIAID. The studies that we've done is looking at the in vitro release kinetics of each one of those individual drugs when formulated alone and when formulated in combination on one of the other APIs, and when formulated as a triple drug IVR. And as you can see from these studies, we are definitely meeting our milestones in terms of releasing the drugs, 100% of the drug within the 30-day duration that we're developing this ring for. But most importantly, from looking at the in vitro release kinetics for all three APIs, there was no significant difference between their release kinetics when formulated alone in combination with one or the other two APIs. So this basically tells us that there is no drug-drug interaction. We're successfully able to formulate all three drugs in a single ring and release within the 30 days. The second most recent MPT IVR that we're developing, this is also funded by NIH and NIAID through an R1 mechanism. And this is being developed to deliver islatravir in combination with the contraceptive combination of NuvaRing, etonogestrel, and ethylene estradiol. So again, we looked at the release kinetics in vitro under different pHs with the intent to correlate with pHs that are relevant to the human vaginal pH, but also the macaque and the sheep pH, given that we were going to test these rings in the sheep and in the macaque. We compared also our rings to the NuvaRing in terms of the release of these two contraceptives. And as you can see, we're releasing on par, if not better, than the release of the NuvaRing with actually less hormones formulated in the ring compared to the NuvaRing. More importantly, and very exciting, is the triple drug IVR with islatravir, showing a very nice zero order release kinetics that's projected to last over 6 months if we continue down this very nice zero order release kinetics. So this actually was designed to release all three drugs. The milestone is to release it within 90 days. So again, we looked at the effect of pH, we looked at the effect of formulating each drug alone and in combination, and we haven't seen any drug-drug interactions either. In terms of in vivo studies, we have done some preliminary pilot studies in collaboration with Dr. Smith at the CDC to look at the release kinetics or the PK of islatravir loaded in these rings, given the fact that this drug has not been delivered topically yet. So as I mentioned, we looked at two different designs, a honeycomb and a solid. We also looked at the difference between the human versus the macaque in vitro before we go in vivo, so that we can correlate the release kinetics in vitro to in vivo. So we miniaturized these rings by keeping the design aspects the same as I mentioned before. And I'm just going to show a couple very preliminary data that we have from these pilot studies, but very highly encouraging. So the graph on the left shows the in vitro data, just to look at how these in vitro data from the release of islatravir from the ring compared to what we see in the plasma. And as you can see, very interestingly, they kind of follow the same trend. So we have this nice high burst at the beginning, and then it kind of plateaus in a zero order fashion or steady state, it hits steady state afterwards. So this was designed as a 30-day or 28-day study. We then removed the ring and collected samples 1 week post removal and quantified the levels of islatravir in the plasma. We also looked at the PBMC levels of islatravir compared to the plasma. The graph on the left is what I just showed, and the graph on the right is the PBMC levels of the EFDA triphosphate, which is the active API or active drug for islatravir. Very encouraging is the fact that we are getting levels in PBMC levels that are above the known efficacious levels post oral dose based on the Merck data. This is just the tissue concentrations versus time. We looked at the vaginal tissues, both proximal and distal. And again, these levels are, the PBMC levels are above the known efficacious levels from the Merck data that we have in hand. So this is very exciting. So just to summarize my talk, we are continuing to develop the 2 MPTs, the one for anti-HIV, HSV, and contraception. We are currently conducting a non-human primate study on that ring to look at the safety and PK. We're continuing to develop the second MPT with islatravir and the NuvaRing contraceptive combo. We have just completed our first sheep safety and PK study, and we will be looking at that data and how it compares to our in vitro data and the initial islatravir data from the non-human primates. And we're also doing safety and for the vaginal microbiome with Rick Piles to look at the effect of these rings on the vaginal microbiome, which is very important in terms of regulatory and moving this technology forward. We are conducting also focus group studies. We have conducted a number of focus group studies, which is a work being led by Dr. Golin in the School of Public Health here at UNC in collaboration with Kate Guthrie at Brown University to look at product user characteristics that are very important to users and which will help us develop the right design and the right product for women who will be using eventually this product as an MPT. So that's the other really advantage of using 3D printing is the ability to prototype these rings and change them based on input from the focus group studies and the women who will be eventually using this. So we've been taking on a lot of these, the information that we've had from focus groups that have been completed already and re-optimizing the designs to implement those likes and dislikes from the women that we've been actually talking to. So this has been a very important part of the work in developing this MPT and the technology in general. So that's what I mean by our generation 4. We've gone through a lot of generations in terms of iteration of design. So with that, as I said, and as Bethany mentioned, my startup company, Anello, is actually developing this technology for a women's health indications, with the first product being a progesterone ring. So we are definitely continuing to move the technology forward, both as an MPT and as a product to tackle women's health indications in general. So with that, I would like to end my talk. Hopefully I didn't take up more than my time. I didn't see the 2-minute paper come up, so I think I'm good. So I'd like to finish my talk by thanking, of course, all the wonderful people that do all the hard work in my lab, but also all the collaborators. Without them, we can't really move the technology forward. It takes a village, and we have really wonderful collaborators at Duke, at UTMB, at the CDC, and at Carbon to allow us to advance our technology using the CLIP technology. Of course, I want to thank all the funding that has been tremendous in supporting our technology and advancing it, from NIAID, from the Echelon Institute for Innovation, from School of Biomedical Engineering. And I would like to thank everybody who signed up today for this webinar, and thank Bethany and for inviting me to give this talk today, and look forward to the discussion.
Wow, that is so fascinating. Thank you so much, Rahima, for preparing this talk, sharing with everyone. We're going to hold off on questions for after Courtney's presentation. And also, just to let you all know, our presenters have graciously agreed to stay on, for an extra 15 or so minutes for additional questions and discussions with everyone. So Courtney, handing it over to you. You're on mute, Courtney.
There, better? Great. Hi, everyone. Thank you for inviting me to speak here today. This is Courtney Jarahian from PATH, and I am going to be talking on behalf of the PATH team about the work that we've been doing on microarray technologies for PrEP as the MPT. So I'll start by giving some background on the MAP technology overall and the work that PATH is doing through our MAP Center of Excellence to advance this technology platform for multiple different indications. And then also give an update specifically on the progress of the PrEP and MPT, the work that's going on. So microarray patches are a drug delivery system in development for vaccines as well as pharmaceuticals that consists of hundreds or even thousands of tiny microneedle projections that deliver a solid formulation of vaccine into the skin. And so there are two main different types. The first are solid coated microarrays, where the projections are made out of a material like a plastic or polymer or metal or silicon and coated with the drug formulation and then dried. And then there are dissolving microarray patches where the actual projections themselves are molded out of a formulation that contains the active ingredient. And so the entire projection itself dissolves once it enters the skin. There are lots of different types of these maps in development and many different designs. Some require applicator devices to ensure the correct pressure and velocity are applied when putting the patch on the skin to make sure all of the projections successfully embed into the skin, while others are designed to be intended to be delivered with just manual pressure with a finger pressing down on them. And then depending on the wear time that is required, many of these also have an adhesive backing to keep them on the skin for the required amount of time before they can be removed. But because these projections are so short, they don't reach the nerve endings in the skin. They're generally less than 1 millimeter long. And so they are generally perceived as either painless or much less painful than giving an injection with a needle. So the particular flavor of maps that is our focus for sustained drug release are dissolving maps. And these are designed in a format such that the needle tips themselves rapidly detach from the patch backing after it's applied to the skin. And so then the backing can be removed after a few minutes, but the tips remain embedded and dissolve within the body to release the ingredient over time. Note this video here is definitely not to scale, but is intended to show kind of the steps that that ensue for the sustained release mechanism. The key advantages of this delivery technology are that it could enable alternative delivery scenarios. So it's particularly for medications that have to be injectable, it has the potential to take delivery out of the hands of healthcare workers and make delivery feasible by either self-administration or by lesser trained community health workers that can be safe and reliable. Because they are potentially more acceptable and easier to maintain, have potential to improve adherence to therapies, and also to reduce the burden on health systems by shifting, task shifting to self-administration and reducing storage and delivery burdens. But this is also a very novel delivery technology, and some of the challenges that remain for this technology as a class are tackling the regulatory approval pathway for this as a new delivery system for regulators and educating them about the technology. The manufacturing hurdles of developing manufacturing methods that are scalable and low cost and high quality for producing different types of maps. And then also understanding the unknown market potential of these technologies for their various different indications. This is just a small snapshot of the wide variety of many different maps that are currently in the development pathway. There are quite a few others that aren't listed here, but just to give a flavor of some of the things that different groups are working on. The most advanced map technologies are in the cosmetics space. So if you are interested in applying a map to treat your facial wrinkles or blemishes or plump your skin, you can buy those now, they're available. There are not yet any approved licensed pharmaceutical applications of maps, but the most advanced product is a migraine treatment map that was developed by a company called Zozana, which is in the final stages of trying to achieve FDA approval of that product, and others that are in late stage clinical trials and could potentially be available in the nearest future include treatments for osteoporosis and diabetes. PATH, as a global health organization that focuses primarily on the needs of women and children in low resource settings, are focused on other applications for MAP that are generally earlier in development, including the contraception and antiretroviral delivery that I'll talk more about soon. And we're also very involved in MAP development for vaccine delivery, including those that are already entered into phase 1 clinical research, such as influenza, measles, rubella, and hepatitis B, as well as many other promising vaccine MAPs that are in preclinical development and working to transition into clinical research, including for COVID vaccines and many other vaccine applications. So this is a very dynamic technology area. There are dozens and dozens of different groups working on different types of microarray patches with many different designs, features, and target uses, including academic groups, small startups, larger companies. And through our Center of Excellence at PATH, we are working to try to communicate and share information across this diverse range of different key stakeholders to facilitate development of the most promising products. So our Center of Excellence was founded with a goal of advancing this technology as a platform for high priority needs, focusing on those needed in low and middle income countries, but also advancing the technology platform as a whole, knowing that there are efficiencies to be gained by working together on this novel technology that would not be as easy to achieve by working on one product at a time. And to do that, we've focused on efforts around engagement and dissemination of information, filling product development gaps, working to understand what user needs are for technology for different applications, and also looking at the manufacturing and regulatory pathways for the technology as a whole and how that can be streamlined, as well as assessing business strategies for different applications to ensure that MAP technologies are sustainable and viable as a commercial product in the future. Some of our key areas of our strategy and work are prioritizing what the global health applications are for the MAP technology platform and disseminating information about user needs for each of those key use cases, as well as evaluating the business case and cost effectiveness for different products, which we've done recently for contraceptive MAPs, looking at the potential business case from a producer's perspective about producing a contraceptive MAP product. In partnership with Cardiff University, we have founded a regulatory working group that consists of representatives from regulatory authorities such as the FDA, as well as pharmacopeal standards organizations, as well as a number of MAP developers from both academia and industry, to work together to identify critical quality attributes and test methods that could apply to this technology as a class and to better define those. And our intention is in the next few months to put out a white paper with the findings of this group that's been meeting over the past couple of years to inform development efforts and also to hopefully influence future regulatory or pharmacopeal guidances and standards that may come out about this new technology class. We've had a key focus on manufacturing, assessing the readiness of MAP manufacturing across the industry for different versions of MAP technology and where there are gaps and need for development of new technologies and equipment. And partnering with Harrow Hoflichter, which is an example of a pharmaceutical equipment manufacturer that specializes in customizing equipment to produce new types of delivery technologies, such as MAPs and many others, to identify what technologies already exist that could be modified for MAPs and where new development is needed. And throughout our work, we aim to disseminate our learnings as broadly as possible through website, through our quarterly newsletter, through conferences, webinars, presentations, et cetera. So now I'll be shifting gears away from the technology as a whole to speak a bit about our particular focus on developing MAPs for HIV PrEP and for contraception as an MPT product. So our approach for this has been to formulate the two drugs separately so that they can either be delivered independently on different MAPs or ideally in the future combined into different segments of the same patch that could be an MPT product providing both PrEP and contraceptive actions. So the unique characteristics of this MAP technology, which is the focus of a partnership that we have with Queen's University Belfast, who has pioneered the development of MAPs that enable a high drug loading. So the majority of the content of the actual dissolving projections is made up of the active ingredient itself, along with some biocompatible water-soluble polymers to form the structure. The target for the MAP for PrEP is a short wear time, either 20 minutes or potentially we can make that less, and enabling a minimum weekly or monthly dosing, although we're strategizing ways to further extend the duration of protection between doses. We're aiming for a product design that could be either administered by a healthcare worker or that would be suitable for self-administration, and that incorporates a feedback indicator technology to confirm to the user that they've applied successful pressure to the MAP to ensure that they have achieved correct delivery themselves. And I'll talk a bit more about the development of that indicator and the work that's gone into that. So this project is a partnership that requires the unique capacities of multiple different groups, including, as I've mentioned, our key collaboration with Queen's University Belfast, as well as working with partners in MAP manufacturing to prepare GMP manufacturing methods for clinical research. And also partnering with organizations in Kenya, Uganda, and South Africa, which are LVCT Health, CHDC, and Genesis Analytics, to conduct in-country assessments with users and key stakeholders to understand usability, acceptability, and programmatic fit of the MAP technology to help inform product development. PATH's approach to product development overall is to take a multidisciplinary approach. We're very keenly focused on user-centered design and tackling questions not only of technical feasibility, but also understanding the broader value proposition of new technologies, including usability, programmatic fit, cost effectiveness, business strategy, to ensure that the technologies that are developed are the right fit for purpose for where they'll be used, and that they will be sustainable in the long run. The work that's being done on MAP design and formulation at QUB includes dissolving microneedles, targeting right now cabotegravir as the antiretroviral drug and norelgestromin as the example progestin for the MPT MAP. Using a layered design where the drug itself is concentrated into the tips with a drug-free backing to the patch. And this increases the delivery efficiency and the percentage of the drug that is delivered, and also enables us to have a tunable rate of dissolving of those two layers to achieve this rapid separation of the tips from the backing to allow for a short wear time, and then also to be able to tune the release rate of the drug tips once they're in the body. And there's been very promising data so far with using the micronized sodium salt formulation of cabotegravir in the MAP in preclinical studies in animals, finding that the pharmacokinetic profile is with that form of the API, which is lower cost and easier to manufacture, is similar or comparable to the PK profile of the injectable long-acting nanosuspension form of cabotegravir. As an overview of the user evaluations that we've achieved to date, we have completed studies in South Africa and Uganda with the goal of exploring perspectives about MAPs, understanding users' acceptability, usability, and programmatic fit of this technology, both for HIV PrEP and then also as an MPT, and then understanding users' needs and preferences for key features of the patch design. And this has very much informed our product development and next steps. And we're working on next generation prototype devices that will soon assess in a similar evaluation with users in Kenya. Some of the feedback that we've obtained from users to date, including a preference for smaller sized maps, but knowing that there is a trade-off between size and duration of protection. Some users would trade a larger size map to be able to achieve a longer duration of protection. The preference for application sites included the upper arm or the thigh. And there was some preference for a short wear time, although many said that up to an hour could be acceptable if that was necessary. The preferences on duration of protection, though, were at least 1 month, but ideally multiple months. So that's something that is a focus of our next development work. In this evaluation, we asked users to compare 2 different prototypes for the feedback mechanism, and both designs are generally acceptable, but there were clearly some design refinements that were needed and to be made in the indicator itself. And many users believe that they would prefer self-administration of the device, although some would feel more comfortable self-administering a product if they were first showed how to do it from a trusted healthcare provider. So the feedback indicator itself is a technology that would be built into the adhesive backing of the patch that would give the user confidence that they have pressed hard enough by providing both a visual and tactile cue that the pressure was successful. And our goals are to make this device easy and intuitive to use, discreet for the user, not obvious what it is, as well as low cost and easy to manufacture and assemble together with the drug-containing microarrays. And so we're working on improving these designs in the next iteration that will for future assessments. And our goal is to make this a scalable size design so it could be applicable to different map sizes and formats and APIs in the future. We've also done some preliminary work on cost effectiveness of maps for PrEP and as an MPT. And at this early stage of development, past focus around cost effectiveness is on the sensitivity analysis side to really understand what the key drivers will be ultimately of cost effectiveness, so that can help inform product development decision making. And so some of the key considerations that we're keeping in mind to keep the overall cost of goods as low as possible included the choice of API, the drug delivery efficiency, the size, both of the overall map and its individual arrays, the duration of protection it provides, and the choice of materials and packaging that are used. And building upon the cost potential range and costs of the product, we also looked at potential impact in service delivery costs, comparing to oral PrEP, and comparing the impact of self-administration use cases and the possibility of increased uptake of a more acceptable product. And some of the key challenges and opportunities that remain in conclusion to this talk. In general, map development so far has focused on highly potent APIs such as vaccines, and that is really due to the dose loading limitations of many map technologies. And so most current ARVs require much larger doses. And so to accommodate that, we've focused on maximizing the drug loading into a microarray, but still trade-offs are going to exist between acceptable patch size and acceptable duration of protection that need to be resolved. And using more potent novel APIs, such as some of those in the pipeline, are likely to be able to address this issue by reducing both the dosage required and the patch size, while increasing the duration of protection that can be achieved. As well as another key opportunity for the technology is to be able to, its ability to deliver a progestin as well, and to combine those two together to better address the needs that we've heard from women, and that I'm sure that this group is very familiar with, that a multi-purpose prevention technology would better address their needs as they see them. And so making that a focus of our development effort to ensure that the PrEP map and the contraceptive map can ultimately be combined together as an MPT product in the future. So a quick thank you to our donors at USAID, PEPFAR, and the FCDO, as well as to all of the project team and partners who contributed to this work. Thank you, and happy to turn it over to questions.
Wow, thank you so much, Courtney. That is fascinating. Both your technology and your Center of Excellence. Thanks so much for sharing all that. So both Rahima and Courtney, fabulous talks. We have been getting some questions in the chat. And Joe Romano, I'm going to hand it over to you to start off some questions if you're available. Otherwise, I'll read off a question from Greg Kopp.
I am here, Bethany, but it's going to be difficult for me to read the chat questions, given that I'm watching on my phone while I'm driving. So I think that should go to you.
Sure.
Okay.
All right. Yeah, so let me, we have, I'll start off with a question from Greg Kopp at FHI 360 for Rahima, asking if your APIs are loaded as solutions or suspensions or combinations.
Yeah, great question. Thank you for that. They are not suspensions, they're in solution.
Thank you.
Absolutely.
Great. Another one from Terry Sin at NIH. Terry, if you're on, do you want to ask that question? It's nice to hear people's voices.
Sure. Happy to ask. I was just wondering, Dr. Benabor, if you could say a little bit more about what the women in the focus groups have told you about some of the initial iterations of the ring and the changes that you made in response to that.
Yeah, absolutely. Great question. And thank you for asking it. I do have to premise that those focus group studies were supposed to all happen in person and had to be shifted to remote because of COVID last year. So just imagine having to, figure out how you do focus group through Zoom. Nonetheless, the team has done a phenomenal job succeeding on holding a number of those. So the things that we were looking at initially were the looks in terms of the color. We really wanted to know what the women responded, how the women responded to different colors in the rings, to see if we needed to pick one color that was a winner. So the colors, it was very skewed, but we definitely honed into three colors that were definitely winners amongst the 8 that we were showing the women. In terms of design, some of the common views from the women that were really common between or reiterated over and over were the size of the designs, whether they were too large. I think that's where some of them really liked it, but most of them didn't like it. And it really went back to them asking how would the design lead to efficacy. And at the end of the day, they really they asked some really good questions in terms of what the design does in terms of efficacy. Was it going to help to have it this big or this small? But in general, the larger designs were not well received. But the smaller ones were very well received. The size of the band, also how much we covered a ring, was one aspect they really gravitated towards more coverage as opposed to less coverage. So those are the things that we really implemented in our second iteration, but we've actually redesigned a ring, and that's something that not only ties into the look, but also the efficacy and how the drugs will be released eventually. So that's something that we're currently working on, designing a new patent around the design technology, and how we shifted from the prior design aspect to the new ones. And I would have to say that the women's input has really been important in terms of comments that they made, in terms of, I really like this one looks like a tire. And so I think those comments were super important in terms of us making them look more feminine, us making sure that the color that we're going to pick is going to be the color that women will gravitate towards, and removing any possibilities for rejection or lack of wanting to use or even start using the designs or the ring itself. So it's still a work in progress. We're just starting our in-person focus groups this summer. So that's very exciting. That way they can actually handle them and they can see the differences between the compression and what the design does in terms of, it's really hard to look at the design through a screen. So it'll be really nice for them to actually look at it and see how smooth they are and how they feel with the designs and without the designs, with more coverage, less coverage, different colors. So really excited to start getting some feedback on that and starting to narrow down our product so we can hone in on one.
That's great.
Yeah, thanks so much. I think it's really exciting to hear in both of these presentations how much these preferred user characteristics are being considered early on in the design process.
Absolutely.
Thank you. I just want to note, I know some people are having to hop off, but this will be, the recording and the slides will be available on the website. So if you go on to the impt.org, on the homepage is a hub for learning events, and that's where all of these are housed. So in another day or so, this one will be up there if interested in listening to the rest of the questions. Yeah, so Joe, I'm going to hand it over to you. I know you had a couple questions.
Thanks, Bethany. And I do have some questions that were previously submitted by others to the IMPT to share today. One of them was, have you considered how to address, oh, sorry, wrong column. What needs are there for the implantable systems to be in the pipeline as MPTs or HIV prevention that you see now? I don't know if that was clear, but. So, and this may be more for the MAP program, since the question is stemming from the point of an implant. And this is not a conventional implant.
That's right. Yeah, it's not a conventional implant, but it kind of works like 100 mini implants, perhaps, if each needle could be considered a tiny little dermal implant. I think some of the key questions that need to be addressed include how the pharmacokinetics that we've seen in animals, how that's going to translate into humans in terms of duration of and release rates. And so getting through the work that we're doing on addressing GMP manufacturing and being able to transition into the clinic to address some of those questions will be key.
So another difference compared to a conventional implant is that once this goes in, you can't take these things out, presumably, right? So do you anticipate addressing the need for oral run-in or concerns over potential long tail with these microarray products?
That's right. Yes, unlike a removable implant, once the MAP is applied, it can't be removed. So it'll be important that there's a good safety database on the API itself before it's administered by MAP. And whether there's a need for an oral run-in, I think depends on what's recommended for that particular API at the point of time when the MAP begins to be used. As for the tail, that's another good key question that remains to be resolved. Like any sustained release technology, there will be some form of tail, and how the kinetics of that tail will likely be different, given it's rather than one large bolus implant from an injectable sustained release product, be multiple tiny depots under the skin. Those kinetics of the release could and the kinetics of the tail will likely be different, and that has to be determined as well on an API specific basis.
Thank you. And a question for both of you. Could you each explain a little bit about your thinking regarding target duration of release that you're trying to get to with your two technologies? Either one of you guys can go first.
I can start. With cabotegravir, which has been the majority of our work so far, what's possible is a week to a month, and that's been consistent with the animal data we've achieved with that API. And so to match that, have tried to focus on the progestin duration of delivery to be able to match that. But I think ideally would be a duration of 3 to 6 months. But achieving that would require use of a new API for sure, one of the more potent ones that are working in the pipeline. So that's something that we're looking at. And I think that that's also a feasible duration to achieve with a progestin delivery system also.
Okay, Raheem, anything to add from the ring side? Are you targeting a specific duration? I understand in the beginning here, you're looking at 30 days, but do you see this technology going for longer than that as per end user need or cost effectiveness reasons? Hello?
I think we might have lost Rahima.
Oh, we lost her? Well, Courtney, you're stuck with answering remaining questions, unfortunately.
Rahima just got back on. So
Okay.
she may not have heard.
I'm so sorry. I don't know what happened. I just got kicked out.
No problem. So the question for you is, what are you anticipating as a target duration of release for these products going forward?
So we can actually tailor them to last anywhere. For the first MPT that we're developing with the triple drugs for anti-herpes, anti-HIV, and contraception, we targeted 30 days with that. With the second one, we're targeting 90 days. And I think there will be like a threshold for MPTs in terms of how long women would want them to stay on, and it really will depend on that input from the women and how long they would want a ring to reside administered and when they remove it. And so with the second MPT being a 90-day on, it also will depend on, of course, efficacy over time. But then once we remove it, what does that tail look like, and what kind of efficacy or PK we would be looking at. But I think in terms of duration, we can tailor them to last a month, 3 months, 6 months, a year, because we have that ability to fine tune the designs to meet those requirements, provided that we can actually achieve, steady state release to the target release kinetics that we're looking at. But really, it all depends on what that duration would be the most accepted by the women who are going to use them.
And presumably for both of you, duration of release will depend on the potency of the drugs as well, as you've both mentioned. So what do you see in the field, where the field is heading in terms of API options? So I think most people on this call know about cabotegravir in the injection. Islatravir is the hot one now. And there's now work going on in non-hormonal contraception. Are there other things you see in terms of API that would enhance the capacity or ability or efficacy of your technologies?
Yeah, absolutely. I can start, and then I can let Courtney add as well. The API's potency is very important, but also because that allows us to not only lower the amount that we would formulate, but also be able to achieve sustained release for the durations that we're targeting with a lower amount of drug. That being said, there's also, besides the potency, there's also the ability to have no drug-drug interactions, so that whatever APIs we're formulating or co-formulating, we want to make sure that they don't interact with each other or change the PK of the other API once co-formulated. So there's potency, there's also the making sure that there's no drug-drug interaction, and how that all ties into the duration that we're looking for, but also the ability to formulate translational doses that are known to be efficacious. So yeah, cabotegravir and islatravir are definitely target drugs that we're pursuing just because of their known efficacy, but also very high potency, some low resistance as well. So I think all that comes in play in terms of what to choose as an API for an MPT development, dependent on the technology. So for the ring, we're definitely looking at those islatravir as our next target.
There's also the advantage as things progress from a treatment perspective that regulatory information would be available for the prevention indication as well. So Courtney, I'll let you take a shot at that with your technology. Thanks.
Yes, agreed that islatravir is a really exciting option, and we're doing some pharmacokinetic modeling right now to understand how long the potential duration of delivery from a MAP could be with that drug, and others in the pipeline, things like lenacapavir or other new agents that are more highly potent, can achieve long duration with a small dose, are also very interesting and exciting. I see also a question from Greg in the chat, whether the rate limiting is due to the API or the map itself, and it can be both. So for APIs that are hydrophobic, like cabotegravir in itself, that produces a rate, a sustained release activity on the formulation, but then also the polymers that are chosen for the microarray patch formulation can also be chosen to provide rate limiting effect as well as the format of the drug that's loaded, whether it's a nanoparticle or other format too. So there's multiple options there to tune the release rate for a more highly potent drug.
Thank you very much. Bethany, since I can't read what's in the chat while I'm driving, I'll turn back to you if there's additional questions you want to share with the group.
Sure.
Thanks to both of you for your excellent discussions.
I do see another question here from Kavita Nanda at FHI. Do you want to unmute yourself and ask a question for Courtney, or I can read it?
Yeah, sure. So my first question was already answered. I think Joe asked the same question. And my second question was for Courtney again. Do you have any animal data? You mentioned the animal PK. Do you have any data that you are able to share at this point on duration of release for either agent? Thank you.
Yes, we do have some animal data in rats, and we're working on a publication of that right now. But there are other presentations that I can forward to that include some of the summary of that rat data.
That'd be great. Thank you so much.
Mm-hmm.
Great. Are there any other questions? If anyone, you can unmute yourself, raise your hand, or just ask any questions before we adjourn. Okay. I want to thank Courtney and Rahima again, just for your excellent presentations. And I know that, these things take a lot of time to prepare. So really appreciate that. And Joe, thank you for facilitating and for, your thoughtful comments and preparation to the discussion. And thank everyone for joining today. It's hard being on Zoom all the time. So we're trying to find ways to connect people. So I wanted to also just show everyone that, there are other resources, that we have available in addition to the learning events. There's some social behavioral resources in the SBR Resource Hub. If you all have other resources that you think would be interesting to share, please let us know. We're also updating the MPT database now too, so that's online and live. And again, if there are technologies or MPTs in development that aren't included in the database, let us know. So with that, I'd like to thank everyone once again, and I hope everyone has a great rest of your week. Thank you.
Thanks everyone. Bye.
Bye.